Welcome to the Longevity Papers podcast.
Hi there. Yeah, it was quite a week for submissions. Really monumental in volume.
But we need to be pretty honest with our audience, right?
Yeah.
Especially for the biotech folks listening.
Absolutely. The signal-to-noise ratio, it was frankly brutal. If you spent your week wading through dozens, and I mean dozens, of preprints on, the usual suspects.
MTOR, SORT-T1, yet another senolytic tweak.
Exactly. Relying heavily on C. elegans, maybe some zebrafish, standard mouse models. Look, that work is solid. It has its place, but it's not where the big shifts, the real model changes are happening. You're not alone if you felt that way.
Right. So we're deliberately skipping the papers that are, let's say, marginal gains this time around.
Mm-hmm.
Our focus today, our mission really, is to cut straight through that noise. We want to talk about 3 specific papers, a couple of preprints, one publication that, well, we think really demand a genuine reevaluation.
Yeah, they could potentially shift how you, as researchers, should be thinking about targeting aging intervention.
We're looking at work from highly credible institutions, obviously, but focusing on mechanisms that are surprising, maybe even disruptive, stuff that might actually require you to adjust your strategic roadmap.
So we've picked out 3 candidates. They span systems biology, epigenetics, and the microbiome. All came out right around October 30th. And the key thing, they seem to move beyond just correlation towards actual potential causal intervention paths.
Okay, let's dive in. Let's unpack these potential game changers, starting with maybe the biggest conceptual shift, the one from Yale.
Yeah, this one's really interesting.
The paper number 1 is titled, Decay in Transcriptional Information Flow is a Hallmark of Cellular Aging. This just landed on bioRxiv, that's BIORXIV. Authors are Emerson, Lin, and colleagues from the Yale University Department of Systems Biology.
And what's really revolutionary, or potentially revolutionary here, is the conceptual frame. They're shifting the focus. Well, instead of just looking at aging as accumulated damage, the classic idea of a machine wearing out, they're proposing it's something more fundamental, a progressive loss of information fidelity.
Information fidelity
Yeah.
within the cell's regulatory networks.
Exactly. It's a systems-level diagnosis, which is pretty elegant, I think.
Okay, that immediately sounds, well, a lot more complex than just measuring telomere length or something.
Yeah.
How do they actually quantify this information decay inside a cell?
Right. That's the core novelty. They used information theory.
Okay. Information theory.
Yeah, they basically modeled the interactions between transcription factors, TFs, and their target genes. They treated that link like a communication channel.
Uh-huh.
And by measuring the mutual information transfer using single-cell RNA sequencing data across 10 different tissues, they could put a number on how certain, how reliable that regulatory communication was.
And across all 10 tissues, what was the trend? Didn't hold up.
It did. Consistently, reliably, that information transfer declines with age. That's the hallmark they mention in the title.
Okay, so information decays. But why? What's breaking down? That's crucial for intervention, right?
Absolutely. And their finding here is key. The decline wasn't because the channel was corrupted, so to speak. It wasn't like the TFs couldn't physically bind anymore. The core problem, they argue, is what they call input distribution mismatch.
Input distribution mismatch. Okay, unpack that a bit. Does that mean just boosting TF levels isn't the answer?
That seems to be the implication. If aging is this failure of communication fidelity, then maybe the current approach is just focused on, say, boosting the overall levels of certain aging-related TFs. Well, maybe they're missing the point if they don't consider the coordination.
Right, so it's not the individual instruments, it's the orchestra conductor.
That's a great analogy. The problem isn't necessarily broken physical parts, it's the loss of that coordinated activity among the TFs. The whole network loses its structure, its robustness, it gets inefficient. Like you said, less like an orchestra, more like a bunch of soloists doing their own thing.
So if the diagnosis is a failure of coordination, did they try to fix it? Restore the conductor, so to speak?
They did explore that, yeah. But importantly, this part is still in silico. It's computational.
Ah, okay. Simulation.
Right. The paper shows that when they computationally simulated upregulating a pretty small specific set of key TFs, they could actually restore youthful levels of information transfer and gene expression, at least in the model.
Okay, in silico success is one thing. Translating that symphony model to an actual in vivo therapy, that sounds like a massive hurdle. What's the single biggest challenge, you think, for a biotech company wanting to pursue this?
Well, there's always the oncogenesis risk, right? Anytime you talk about upregulating transcription factors, especially broadly, cancer risk is the elephant in the room. But beyond that specific risk, the main challenge here is identifying the consensus set of TFs. They found specific ones that worked in silico for specific tissues, but do those overlap? Is there a core, safe, maybe universal set of master regulators that can fix this network failure across different tissues? 10 tissues in their study. That's a huge validation effort needed in vivo.
Finding that master conductor, or maybe a small committee of conductors.
Exactly. And then figuring out how to deliver them safely and effectively, AAV, lipid nanoparticles, whatever it might be.
Okay, so if the Yale paper gives us this high-level systemic view, aging as a loss of coordination, maybe we need to find the specific players, the specific conductors whose absence really causes problems.
Which leads us nicely into the second paper.
Yeah, this one seems to drill down into a really specific mechanism for maintaining control, especially epigenetic control.
It's a really beautiful piece of mechanistic work, I thought. It connects DNA repair, epigenetics, and senescence in a very direct way.
This is the one titled, FOXM1 enhances DNA repair in aged cells to maintain the peripheral heterochromatin barrier to senescence and answers.
Yeah.
Also popped up on bioRxiv, October 30th. This comes from I3S, the Universidade do Porto.
Right, and the focus is FOXM1. Now, FOXM1 often gets linked to proliferation, cancer.
The oncogene connection again?
Yeah, but here they're identifying it as something else, a critical age reversal factor. They're arguing it preserves genetic and, crucially, epigenetic integrity. It seems to actively inhibit the senescence program before it fully kicks in.
So more about prevention than clearing out senescent cells later.
Exactly. Holding the line, not just cleaning up the mess.
Okay, let's walk through the pathway they propose. It seems quite intricate, quite elegant. How does FOXM1 actually do this? How does it hold the line?
So it seems to start with FOXM1 levels declining with age, which is known. This age-related repression leads to a sort of transcriptional shutdown of key DNA repair genes.
Okay, step one, less FOXM1, less DNA repair.
Right. So step 2, if you restore FOXM1 activity, you enhance DNA repair efficiency. Now, this repair efficiency is critical for the next step. It prevents the degradation of an enzyme called G9A methyltransferase.
G9A, that's important
for maintaining a specific epigenetic mark, HQK9E2. And G9A seems susceptible to degradation when there's DNA damage.
Got it. So FOXM1 boosts repair, which saves G9A, and G9A stability is key
because G9A maintains that H3K9E2 mark, specifically at the nuclear periphery. Think of it like building an epigenetic wall or barrier there made of heterochromatin.
Okay, a barrier at the edge of the nucleus.
Exactly. And maintaining that barrier is what silences the enhancers for genes involved in the senescence program, particularly the inflammatory stuff, the SSP, driven by AP1.
Wow, okay. FOXM1, right hero DNA repair, right hero G9A stability, right hero peripheral H3K9E2 barrier, right hero stasis P silencing. That's quite a specific chain of events.
It is, and while it definitely builds on known senescence biology, people like Campisi, Van Dersen have laid groundwork here. This particular linkage, this specific sequence, feels highly novel and very mechanistic.
It's undeniably a powerful mechanism they've described, but strategically, for someone in biotech, you immediately hit that FOXM1 problem again, don't you? The oncogene risk.
Absolutely. It's the major translational dilemma.
So if G9A stability, maintaining that H3K9E2 barrier, is the really critical downstream step, why focus upstream on FOXM1 at all? Is the smarter play here maybe to bypass FOXM1 entirely? Could you target G9A stability directly with small molecules, perhaps?
That's precisely the strategic crossroads this paper puts us at. They've established FOXM1 as this upstream linchpin, connecting the DNA damage input to the epigenetic defense output. But you're right, translationally, the path of least resistance, or maybe least risk, might involve decoupling that defensive function from the proliferative oncogenic drive, the high-risk, high-reward path, maybe a super-specific FOXM1 agonist that only affects the DNA repair genes, not the cell cycle ones. That's a tall order.
Sounds almost impossible. And the safer, maybe more actionable route.
Well, like you suggested, maybe small molecule screens specifically aimed at stabilizing G9A, or finding compounds that enhance H3K9EME2 maintenance just at that peripheral barrier that addresses the vulnerability they identified, potentially without poking the FOXM1 bare.
Right. This paper gives a strong rationale for that kind of targeted screen.
Definitely provides clear functional evidence for targeting G9A or that barrier maintenance.
Excellent. Okay. So we've gone from a systems-level problem information decay down to a potential specific molecular mechanism involving FOXM1 and G9A for maintaining fidelity. Let's pivot one more time. Paper number 3 connects the outside world, specifically the gut, back into these core nuclear processes like DNA repair.
Yeah, and this one's exciting because it feels immediately, well, more translatable. It offers a clearer path, perhaps, and validates this idea of reinforcing these failing regulatory points.
So this is the paper titled Lactobacillus salivarius-derived indole-3 acetic acid promotes AHR-PRP1 axis-mediated DNA repair to mitigate intestinal aging. This one was published October 28th in Advanced Science. Authors Cao, Zhang, and colleagues from the Chinese Academy of Sciences in Wuhan.
Right, CIS Wuhan.
Now, indole-3 acetic acid, IAA. We hear a lot about microbial metabolites activating the aryl hydrocarbon receptor, AHR. What makes this particular IAA-AHR PARP1 link stand out? Is it just another metabolite receptor story?
That skepticism is fair. There are tons of papers linking metabolites to AHR, but I think the novelty here is twofold: the specific context, intestinal aging, and the mechanism they uncover.
Okay, tell me more.
So first, they show that this specific metabolite, IAA, derived from a common gut bacterium, Lactobacillus salivarius, is actually quite effective at mitigating signs of intestinal aging. Things like DNA damage, gut barrier dysfunction. That's a really critical aging phenotype that often gets overlooked but drives systemic issues.
And the mechanism, how is it distinct?
So IAA does activate AHR, as you'd expect, but here's the twist. The activated AHR doesn't just act as a standard transcription factor, turning genes on or off. They show it physically interacts with PRP1.
Interacts with PRP1, the DNA repair enzyme.
Yes. And it potentiates its activity. It actually enhances pyrrolation and DNA repair directly. It's not just a transcriptional effect. It's a direct functional boost to the DNA repair machinery itself via this AHR-PRP1 interaction.
PRP1, which is, of course, smack in the middle of NAD plus metabolism and genome stability debates in longevity.
Exactly. So this connects that whole established NAD plus PRP1 longevity territory directly to the gut microbiota.
Wow.
It suggests a potential, almost natural pharmacological way to shore up a crucial regulatory node, PRP1 activity, that we know falters with age, especially in a high-stress place like the gut lining.
And the translational path seems pretty obvious. Supplement with L. salivarius itself, or maybe even just IAA.
That's the immediate thought, yes. But of course, there are risks and questions for any researcher wanting to move this towards the clinic.
Such as?
Well, specificity and safety, as always. First, you need to make sure that boosting this pathway doesn't inadvertently help out pathogenic bacteria that might also use IAA, or somehow disrupt the gut microbiome's overall balance in a negative way. That's always a concern with microbiome intervention.
Right, unintended consequences.
And second, AHR activation itself can be a double-edged sword. In immune cells, for instance, it can sometimes be pro-inflammatory. So you'd need really careful validation that this specific IAA-driven AHR activation and its interaction with ARP1 stays beneficial and localized primarily to the intestinal epithelial cells, giving you the anti-aging DNA repair boost without causing unwanted systemic immune effects.
Ensuring the fix doesn't create a new problem elsewhere. Makes sense. Okay, let's try and pull these threads together. It feels like this week, despite the noise, really offered more than just data points. It gave us some new frameworks.
I think that's the key takeaway, yeah. The really impactful work seems to be unifying fields that were maybe a bit siloed before. You know, we saw systems biology meeting rejuvenation factors in that Yale paper.
Epigenetics tying directly into DNA repair with the FOXM1 work.
And the microbiome talking directly to nuclear signaling and repair in the CAS Wuhan paper. It's about integration.
And for you, the biotech researcher listening, the strategic thread seems to be this shift in focus, moving away from just trying to, slow down the decline.
Towards actively trying to reverse damage or dysfunction by restoring coordination, restoring that information fidelity we talked about earlier, restoring fundamental cellular integrity, whether it's information flow, chromatin structure, or gut barrier function.
Right, it's a more proactive stance.
Precisely. So maybe the provocative thought to leave everyone with is this. If that Yale model, paper 1, is right that aging is fundamentally a loss of coordinated TF activity that can be corrected by boosting key players, then is FOXM1, the factor maintaining that crucial heterochromatin barrier in paper 2, potentially one of those specific coordinating TFs the Yale paper is hinting at least for the epigenetic and DNA repair side.
Connecting paper 1 and 2.
And then could that microbiota-derived IAA from paper 3 be acting as a sort of natural, targeted drug delivery system, a way to reinforce precisely those key regulatory nodes like the PRP1 repair hub that are failing in vulnerable tissues like the aging intestine?
So using the microbiome to bolster the specific weak points identified by the other mechanistic work.
Could be. That potential synergy, linking systems, failure to specific molecular weak points and then to external modulators. That feels like where the really exciting next steps might lie. That's the connection that demands attention now, I think.